The first one will be Maria van Korff. She is a professor at the University of Düsseldorf. I actually find it an honor to introduce you, Maria, because before I've been working as a postdoctoral researcher in Maria's lab. And Maria's main topic is on looking how we can make barley more prone to stress conditions, for example, high ambient temperature. And then the second speaker we will have is Gesa Helmsorig. She's a PhD student in Maria's lab, and she will be soon finishing her PhD. And she mainly worked on flowering locust tea. But first, we're going to listen to the seminar of Maria. So without further ado, Maria, let's hear your research.
Okay. Thanks, Vilma, for the introduction, and thanks for inviting Gesa and me to present some of our work today. So as you said, I will talk about barley inflorescence development, actually both of us, and I will talk a bit more about development under high ambient temperature conditions, and Gesa will explore more the genetic control of inflorescence development. Now, trying to see if I can forward my slide. No. Okay, so you all know global warming threatens crop production, and it is forecasted that really there are huge yield losses due to, in particular, temperature raises, but also changes in precipitation. And this is just here shown for wheat across different countries and globally. And it's in particular reproductive development that is impaired under high ambient temperature conditions and or very susceptible to temperature variation. And what actually happens is that you see typically a strong decrease in the number of seeds formed per spike, as I'm showing here in the slide, where you see one beautiful barley spike, 2 rows under controlled conditions, and on the right-hand side under high ambient temperature conditions when there are only very few grains formed. Now, many years ago, we started off by trying to see or screen natural genetic variation to see if there is variation in reproductive development in response to temperature or ambient temperature variation. And for this, I made use of a reference collection that I was involved in putting together when I was working at ICADA under the Generation Challenge program, when a large collection, about 30,000 barley gene bank accessions were screened. The core sample was put together based on passport data and origin, genetic origin or geographic origin. And then this core sample of about 3,000 accessions was genotyped at that time, still a bit more classical genotyping with markers. And then we put together a reference collection, about 300 accessions to maximize genetic diversity. And this collection I took back to Germany, single seed propagated this collection and used it for various things. And today I would just like to talk about a subset of this collection, about 100 genotypes. Many of them, or most of them, were landrace or are landrace accessions from Northern Africa and the Middle East. And then we added a number of cultivars, in particular from Middle Europe and Northern Europe. And we scored these lines, or just here maybe to see, so the landrace collections we selected to really come from areas with very high mean maximum temperatures to see how they would be doing in response to high ambient temperatures compared to our cultivars from Europe. And we also genotyped this collection with a 50K SNP array. We scored the collection under controlled conditions, where we grew them first under short day conditions, and then we shifted them to vernalization because most of the landrace genotypes are winter types, so they need a period of cold to flower. And then we grew them either under control, so what we call control conditions. And let me see if I can find my pointer. So and that would be 20 degrees during the day and 16 degrees during the night under long day conditions, or under 28 degrees during the day and 24 degrees during the night. We scored a number of vegetative traits during development and a number of reproductive development at harvest. And today I just want to focus on a few traits. Actually, flowering time, that is one of our favorite traits. And what you can see here in this graph, a bit difficult to see, but I just will show you through. On the x-axis are the different genotypes and accessions. And on the y-axis, you see the number of days after vernalization to heading or flowering time. And there was huge variation in flowering time, but then we could see there was a set of genotypes that really accelerated flowering time under high ambient temperature conditions. So in blue is control, and in yellow is high ambient temperature conditions. So they were earlier under higher temperatures. And then there was a set of genotypes that got strongly delayed under high ambient temperature conditions. And some of them didn't even flower. And it wasn't completely random how these accessions were distributed. So here, those that accelerated flowering were mainly landrace genotypes from hot environments, and those that delayed flowering time or could not really flower were mainly elite barleys from Europe. Now, rather than to show you all the other traits, I give you a summary. So what I'm showing here is the correlation of a number of traits that you see here on the x-axis to seed number. And in blue, again, control, and in yellow, high ambient temperature conditions. And if we look at, we call maybe vegetative traits, so leaf size, biomass, leaf number, we could see that these were positively correlated to seed number under control conditions, but negatively correlated under high ambient temperature conditions. And if we consider what we call maybe yield component traits or traits that directly contribute to seed or grain number, then we could see that, So what we can see here, so this is on the right-hand side here, you see a spike that is developing or an inflorescence. And this inflorescence here forms these spikelets, and each spikelet can then develop into a floret. So what we saw is that the spikelet number, so the ability of the inflorescence to induce many spikelets, was positively correlated, or highly positively correlated to final grain number per spike under control conditions, but that wasn't the case under high ambient temperature conditions. In contrast, under high ambient temperature conditions here in yellow, it was not the number of spikelets, but the ability of the spikelets to form a floret and the ability of the floret to really form a grain. So there were different components that really contributed to the final grain number. And you could also see that there was a different correlation of a number of vegetative traits to final grain yield here. So correlation of traits really to seed number or grain number really depended on the environment. Now then we used the SNP data to look for correlations or for associations, and we found one major gene that was controlling variation in flowering time in response to high ambient temperature conditions. And that was a gene that we already knew. It's the major photoperiod response gene, PBDH1 in barley, that is an ortholog to a pseudo-response regulator gene from the circadian clock in Arabidopsis. And we know in barley that this gene has already been shown by the group of David Laurie, that this gene under long day conditions in the leaf induces the expression of a barley ortholog of flowering locus T that, as we know from ricin, Arabidopsis moves as a protein from the leaf through the vasculature to the shoot apex to induce inflorescence development. Now, in barley, there is a natural variant that is known, and this is a SNP that causes an amino acid change in the CCT domain of this gene. And this is really what we call a hypomorphic allele, because it reduces the functionality of this gene. It's not abolishing the function. So lines that carry this variation or this variant, they still induce flowering, but the induction of FT1 in the leaf is lower and is delayed, and that causes then a delay in flowering time and a delay in inflorescence development. Now that had been described, but then we wanted to understand how does this gene control flowering time in response to high ambient temperature conditions? And if we look into the population, then we saw that if we divide the population at this variant, so those lines that carry a G here at a SNP carry the wild type PPDH1 allele, and under control conditions, they are early flowering, as had been described before, as compared to the lines that carry the SNP. But then what was interesting is to see that the lines with the wild type allele, they further accelerated flowering time under high ambient temperature conditions, whereas those that carried the mutation delayed flowering time. So what we saw here is something like a crossover interaction of flowering time at PPDH1 in response to ambient temperature variation. Now we wanted to dissect this a bit more, and rather than use the large population, we used a shortcut, and that is we looked at integration lines. So we generated an integration line, and the background of Golden Promise, which is a spring barley that some of you may know because it's typically used for transformation. And we integrate the wild type PPDH1 allele from the winter barley, Igri. And because this line then was early flowering, also under controlled conditions, we termed this line Golden Promise Fast. And for everything that we did then with this line, we also did, we had two additional independent integration lines in the background of Scarlet, another spring barley, and Bowman. And here we integrate the white, or that was not done by us, but by colleagues. So the winter barley, or the wild type PPDH1 allele, was integrated from wild barley here. So very different origin of the same allele of PPDH1. So when we grew these lines under control on high ambient temperature conditions, we saw very similar response of both genotypes in terms of growth. So they were shorter, both of them, and biomass was reduced. But then when we looked at flowering time, the response was different. So Golden Promise here on the left, very much delayed flowering time. So again, green is control and yellow is high ambient temperature conditions, whereas Golden Promise Fast accelerated flowering time. So we could recapitulate this crossover interaction effect of PPDH1 that we saw in the population here in the integration line. But then, so that was flowering time, but we wanted to understand how does the whole of the inflorescence development actually behave under high ambient temperature conditions. And for this, what we have to do is we have to dissect the plant as it develops to then score the development of the main shoot apex, which we can do by using the Wadding scale, which really scores the development of the carpel of the most advanced floret on the spike, which is typically here in the middle, in the central part of the spike. And there are two, for us, interesting stages, so this is the Wadding stage 2, which really marks the induction of spikelet primordia. So this is what we call the double ridge stage, when we say there is a reproductive inflorescence that is formed, spikelet induction starts, and then the stage 3.5, which is the anther primordium stage, when floral development really kicks off and the meristem starts growing upwards in the stem. So that is coinciding to more or less with stem elongation. So here spikelets start to be induced, and they are being induced until around stage 4.5 to 5 or 5.5, when then not all of these spikelets form a floret and a grain, but there is a process called floret abortion that kicks in, and that varies depending on the genotype and the environment. So what we are really interested in is understanding what is the effect of high ambient temperature on the activity on the inflorescence meristem here at the tip. So barley is indeterminate, has an indeterminate spike, so this inflorescence meristem remains alive, and then it really initiates triple spikelets meristems on its flank, which then form these spikelets here. And then two-rowed barley, as you see here on the right-hand side, it is only the central spikelet that forms a floret and a grain. So then what really counts is how many spikelets are induced on the inflorescence and how much are these spikelets able to form a floret and a grain. So that determines then the final grain number on the spike. Sorry. So, and what happens if we score, so dissect the inflorescence meristem over development? You can see here in Golden Promise and Golden Promise Fast, again under control and high ambient temperature conditions. And what we observed is that there wasn't a huge difference in development until about stage 3.5, when floral development starts, when then really under high ambient temperature conditions, this floral development was very much delayed and altered in Golden Promise. But that was very different in Golden Promise Fast, where floral development got accelerated. So that showed to us that high ambient temperature had a main effect on floral development, and it either delayed floral development or it accelerated floral development depending on the genotype. And then what we could also do is to score the rate of spikelet induction in response to high ambient temperature conditions. So how fast could the EM induce spikelet primordia here on its flanks? And this you see here for Golden Promise, in green here again, control. So this is very rough, but on average 1.7 spikelet meristems were induced per day under control conditions, and that was strongly reduced by half almost under high ambient temperature conditions in Golden Promise. And that looked very different in Golden Promise Fast, where actually it got accelerated. So the induction rate was the same more or less in both genotypes under control conditions, but under high ambient temperature conditions, this rate got accelerated. What you can also see here is that not only the rate was affected, but also the duration of spikelet induction. So both of them were affected by high ambient temperature conditions and the allelic state at PPDH1. Now we know that it's the inflorescence meristem that is important for the spikelet induction. So then we looked at changes in the morphology of the EM under high ambient temperature conditions, just very roughly by looking at changes in the height and width. And here you see changes in the length of the inflorescence meristem at 2 stages, again in both genotypes. And we observe that so from the beginning of spikelet induction to the beginning of floral development, there was a strong decrease in the length of the inflorescence meristem in both genotypes, and that was very much comparable. But then what we observed in Golden Promise is that under high ambient temperature conditions, there was a strong decrease in the length of the EM, and that was true at both stages, whereas in Golden Promise Fast, we couldn't see a strong difference in the EM size. So that is something we have to further explore, but it suggests that that could be related to the differences in the ability to maintain the activity of the inflorescence meristem in response to high ambient temperature conditions. Now, when we looked at the final spikes that you can see here in Golden Promise and Golden Promise Fast under control on high ambient temperature conditions, we saw big differences in the length of the spike. And that had to do with the difference in the rate and duration of the induction of spikelet primordia, and of course, the developmental timing of this between Golden Promise and Golden Promise Fast. So many more florets were formed in Golden Promise under control conditions, but then there was a strong drop under high ambient temperature conditions. And fewer spikelets are induced in golden promise fast, typically under control conditions, but then the difference wasn't very big under high ambient temperature conditions. But we can see here actually that it's not only the size that is different, it was also the number of seeds formed. And here in particular, golden promise, we could see a strong drop. So there in the number of grains per spike, so that under high ambient temperature conditions, golden promise formed fewer seeds or grains than golden promise fast under high ambient temperature conditions. And that then led to a strong drop in florid fertility. So this is the number of grains formed per florid in golden promise, but not so much in golden promise fast. So for the final seed number, it was really the florid fertility that mattered most. And then we were wondering, why do the florids not form a grain under high ambient temperature conditions? And to test this, we did cross-pollination experiments where we either grew the ovary at 28 degrees and used pollen from 20 degrees to pollinate, or we grew the ovary at 20 degrees and used pollen at 28 from 28 grown plants to pollinate. And this is what you see here. You see a spike that was grown under 20 degrees, and then we pollinated here on this side with pollen from 20 degrees, and on the left-hand side with pollen from 28 degrees. And you see here the strong difference in the seed set that was quantified for actually two different sets of integration lines. So this is always the spring barley here, and these are the two integration lines. And you can see that it was primarily when the pollen came from 28 degrees, here in light blue, in the spring barleys, when there was a strong drop in florid fertility. But also when the ovary was grown at 28 degrees, there was some decrease in fertility, which was not so much observed in these integration lines, which is this one and this with the capital PPH1. And when we looked at pollen shape, what was interesting is that the pollen in golden promise under high ambient temperature condition often looked completely shrinkled and emptied and was not viable based on Alexander staining, and that wasn't so much the case in golden promise fast. What was also different was the anther development, which was clearly impaired in golden promise. So you see here the fertile anthers in yellow under control conditions, whereas they were much shorter and they were only pale and had very little pollen under high ambient temperature conditions. And that wasn't the case in golden promise fast. So what was also altered was where the seeds, so the seed set was changed, but also the seed shape was changed. And you see here the seeds in golden promise control in high ambient temperature conditions. I'm not sure if you can see it, but really the seeds were much slimmer and longer under high ambient temperature conditions, in particular in golden promise, but also to some extent in golden promise fast. And this is what you can see here. So there was an increase in the seed length under high ambient temperature conditions in both genotypes, but there was a decrease in seed width, in particular in golden promise, and to a lesser extent in golden promise fast. So then we've now started to explore whether along with the seed shape, also the seed composition is changed in response to high ambient temperature conditions. And this is some preliminary results where we can see that indeed that is the case. So we can see an increase in nitrogen in both genotypes under high ambient temperature conditions, and then also in the carbon-nitrogen ratio, which is reduced under high ambient temperature conditions in both genotypes. But we could also see some changes in micronutrients such as iron and zinc, which are affected apparently by high ambient temperature conditions. And this is something that we need to further explore. So while we see in lots of phenotypes that are changed, we wanted to also explore a bit more what happens on the molecular level in the inflorescence. And for this purpose, we did RNA sequencing of developing inflorescence meristems, so at the vegetative stage, then at spikelet induction, at the anthrapymortem stage, and then during rapid spike growth. So this is stage 6, where most of the spike growth happens, and also most of the floral abortion. And we did this again under control and high ambient temperature conditions. And we also looked at leaves alongside. And what we were interested in is understanding what is like the overlap of genes that are developmentally important, so developmental regulators, and to what extent are these temperature regulated. And there we found about 500 genes, so developmental genes that are temperature regulated. When we looked at the overall expression changes, what was striking is that we found many more genes differentially regulated at these 3 first stages in Golden Promise than in Golden Promise Fast. So red here is those that were significantly up, and in blue significantly down regulated. So there were many more in Golden Promise. And when we looked at these, so these were heat shock proteins. So again, here the dashed lines. So the yellow is Golden Promise, the blue is Golden Promise Fast, and the dashed line is the high ambient temperature condition. So we found a number of heat shock proteins, particularly upregulated in Golden Promise, but also then quite a number of histone-related genes downregulated specifically in Golden Promise under high ambient temperature conditions, and many genes involved in cell cycle regulation. And so these are genes that were generally regulated by temperature, not necessarily involved in development. So this suggested that although we could not really see a huge difference in the overall phenotype between both lines under high ambient temperature conditions on a molecular level, we could actually identify many quite different responses of these two different genotypes. And then rather than to go into all the developmental genes, I'm taking here shortcuts. So by using information that we know from literature. So when we think about the development of the inflorescence, then we know there is a set of genes that is important to maintain inflorescence development. And there is a set of genes that are important for differentiation and floral development. And what is in the inflorescence really important is the dosage of, or the balance of, flowering locus T that I already introduced. And a close relative of this gene is terminal flower 1, which is not an inducer of floral development, but is really repressor. So the dosage of the two is known to control the vegetativeness or the reproductiveness of a plant. Now in barley, of course, we know, so there are homologs, and that is another FT-like gene is called FT2, and centroradialis, which is a TFL1 homolog in barley. And of course, as already introduced, we know that PBH1 is important for the induction of FT1 in the leaf, but then presumably moves to the inflorescence and induces this switch from a vegetative to reproductive meristem. So when we looked at FT1 expression in the leaf, we could indeed see that high ambient temperature conditions, or just to start off with, there's a big difference between Golden Promise in yellow and Golden Promise Fast in blue, as that was already shown. So that's dependent then on the variation of PBH1. But then under high ambient temperature conditions, we could see that in both genotypes, FT1 is downregulated. And that downregulation then actually correlates to a downregulation of FT2 in the meristem. So FT2 is expressed in the meristem. So we see here a strong response of FT-like genes, which we know are downstream of PPDH1. Then, of course, that, maybe to go back, that did not so much correlate with this acceleration of development that we saw in the line in the Golden Promise fast under high ambient temperature conditions. So then we went into the next stage, which is this floral development stage 6. And we looked at a number of developmental genes. So from this ABC model genes, so for example, Cipolata-like genes. And here we saw, again, this crossover interaction effect. So where we see a downregulation in Golden Promise and an upregulation of Golden Promise fast. So there were a number of ABC-like genes that showed this pattern that does actually correlate with this difference in development in response to high ambient temperature conditions. But there were a number of other genes that showed a very similar expression pattern, and these were, for example, genes related to sugar breakdown, also cytosolic invertase, which is important for the sugar breakdown, and also trehalose 6-phosphate, where we know that trehalose is, at least in Arabidopsis and in rice, is a signaling molecule that signals the sugar state of the plant. So these were strongly downregulated in Golden Promise and were clearly upregulated in Golden Promise fast at this stage. And in addition, there were a number of auxin-related genes, which we see here, a number of these that were specifically regulated in a very similar manner, so downregulated in Golden Promise and upregulated in Golden Promise fast. And we started to explore this a bit further by using reporter lines, and this is what you see here. So this is a reporter line pin 1, which is important for polar auxin transport. And we cross this line, so we have this line in the background of Golden Promise and Golden Promise fast, and image that under control and high ambient temperature conditions. And what you can see is that there was a strong downregulation of the signal under high ambient temperature condition in Golden Promise, and much less so in Golden Promise fast. So here's the imaging data, really. So this is something that suggests that auxin homeostasis or transport is really changed in response to high ambient temperature conditions in a genotype-dependent manner. And we are currently exploring this further by using a DR5 reporter line, but there we don't yet have results. So this is something for the future. So here, I think I've given you some idea what happens under high ambient temperature condition to inflorescence development. And because we've seen that FT-like genes seem to be quite central, I will now, first of all, I should really thank, I have to say, Tian Yulan, who's probably also in the audience. So she has really generated most of the data that I've presented today. And there were other people involved, of course, here, collaborators I would like to thank. But because FT-like genes seem to be so central, now Gesa will take over and talk a bit more about the function of FT1 in inflorescence development. So thank you, and I'm stopping, and I think Gesa can take over.
Yes, thank you.
Thank you, Maria, for a nice presentation. And then Gesa, I would say the floor is yours, right?
Yes. Can you see my screen full screen?
Yes.
Sorry?
I don't see anything.
You don't see anything?
No.
We can't see anything just yet.
1 second.
Yeah.
You can see it now?
Yeah, there it is.
Okay,
Yeah.
great. Let me get things sorted. Now it's in full screen, right?
Yes.
Okay, great. Right, so thanks a lot. Thank you for also giving me the chance to talk here. As Maria already mentioned, from now on it's going to be not so much about heat stress, but instead I'm focusing only on a single gene, which is flowering locus T1, and what effect it has on barley development and also on plant architecture. So an important step in a plant's development is basically going from what we see here on the left, from vegetative growth to reproductive growth here on the right. And the step is generally highly regulated by internal but also by external factors, such as, for example, photoperiod. And we already do know that flowering locus T is a central hub in coordinating the induction of flowering. And the role of FT is actually highly conserved. So we can see a similar process in long day and also in short day plants. So the examples I'm showing in this very rough overview here is on the left Arabidopsis, and on the right we have rice. On the left it's called FT, and on the right in rice we also have an FT-like protein, which is called heading 3A. But the whole process is very similar. So in both plants, we have in the leaf several genes, including, for example, clock genes that upregulate the expression of FT1. And then the produced FT protein is then moved from the leaf through the phloem to the shoot apical meristem, where it can induce the flowering process. And when we come to barley, this process is very similar. So once again here, we have flowering locus T1 in the leaf, and its expression, as Maria mentioned before, is upregulated by different genes, and a major one of them is called photoperiod 1. And we have these two different alleles of PPDH1. So we have the wild type allele that strongly induces the expression of FT1 in long days. And we have the mutated version, which also upregulates FT1, but not as much, and also later during development. And this results in this delay of development and flowering in these plants. And after that, the FT1 protein is also moved from the leaf to the shoot apical meristem, where it can then interact with downstream genes such as florihomeotic genes and induce this change from vegetative to reproductive growth in the inflorescence. So we do already know quite a lot, but what we don't know is what genes are actually controlled by FT1, and also what effects FT1 has on development and also on plant architecture. And this is what my PhD project focused on. So to come to the plants that I worked with, these are the same ones that Maria already introduced to you. So we have the spring cultivar Golden Promise here on the right, which has a mutated PPDH1 allele. And on the left, we have the introgression line with the wild type PPDH1 introgressed into it, which we called Golden Promise Fast. And because we wanted to see the what effect FT1 has on development, Jochen Kumlin from the IPK and his group created knockout mutants for us using CRISPR-Cas9. And with this, we have 3 different FT1 mutant lines, which have a non-functional FT1 protein, which are called FT1A and FT1B in the background of Golden Promise Fast, and FT1C in the background of Golden Promise. And these are also the colors I will be using throughout the rest of my talk. So let's start with the most obvious phenotype we could see in these plants, and this is biomass and also an increase of the lifespan of these plants. So on the left here, we have a Golden Promise Fast plant, which is 2 months old. It has already flowered, and we can see the spikes are emerged and during grain filling. And after 1 or 2 more months, this plant will be fully dry and its life cycle is over. And in comparison to that, we have next to it an FT1A mutant plant, which is 10 months old at this time point. And we can see this plant is still alive, even though some of these tillers have already fully developed or stopped development and are already dry. but this plant still produces fresh tillers. So it continues to tiller even after this time, and it actually continued to tiller for at least 1 more year. And after that, I actually discarded the plant due to space reasons, and it might have even continued to grow after that. So the lifespan of these plants is a lot longer than what we usually see. And an effect of this is also visible in the tiller number, which I've plotted here on the right, which you can already see in the image here. So the tiller number is significantly increased in these FT1 mutants in comparison to their parent plants. When we now look at the flowering time, if we just look at Golden Promise Fast and Golden Promise here, we can really nicely see this PPDH1 effect. So because these plants were grown in long day conditions, we see that Golden Promise plants flower significantly later than the Golden Promised Fast plants. But we can also see that all 3 FT1 knockout mutants here flower significantly later than their parents. Actually, some of them never flowered. And we can also see that we don't see any differences between these 3 mutants in flowering time, regardless of their PPDH1 background. And this lets us presume that this effect on flowering time, this PPDH1 effect on flowering time we see, is conveyed exclusively through FT1. Now, to look at the development a bit more in detail, I also come back to this Wadding scale, which was already introduced to you. So just as a very quick reminder, we use this to score the main shoot apex development, and we start at Wadding stage 1 at vegetative growth, continue Wadding stage 2, where we have this change from vegetative to reproductive growth, and it goes until Wadding stage 10, where we have the pollination. And during this reproductive stage, we can divide this into early and late reproductive development. And in particular, this early reproductive development is very interesting and important for the yield of the plant, because this is when spikelets are initiated, and only spikes that are initiated have the potential to develop into florets and then later into the seeds on the spike. So when we look at the development of these plants, we have here the Waddington stage plotted against the days after emergence. Once again, here in these gray lines, we can very nicely see this effect of PPDH1. So the Golden Promise plants are delayed during all developmental stages compared to Golden Promise Fast. But when we now look at the mutants again, we once again can see this huge delay in development in comparison to the parents. We can see this from a very early stage on, so already before they transition to reproductive development, we see a delay in development in these plants. And we can also see that once again, there are no differences in the developmental speed between these 3 lines. And as I just mentioned, especially this early reproductive development is particularly interesting because this is when spikelets are initiated. And as you can see here, this phase is a lot longer in the FT1 mutants compared to the wild types. So these plants have a longer period of time where they can initiate spikelets on the developing inflorescence. And this is actually also what we can see. So first, we once again have here this difference between Golden Promise Fast and Golden Promise. But we can also see that in all 3 mutants, the number of spikelet primordia is significantly increased in comparison to the parents. The inflorescence size itself is not that much increased, but the spikelet primordia number is higher because the density of spikelets that are initiated is higher than in the mutants. So in theory, these plants do have a much higher yield potential than the parents do, but there is, of course, a downside. So this is very similar to what Maria already showed about these heat stress plants. When we look at the developing floret and we look at the anthers here in Golden Promise Fast, we see that they are green or already turning yellow, so meaning that they are very close to pollination. And in comparison to this, the anthers in the FT1 mutant look very small, very pale, and don't really seem to have a lot of pollen. And also here we did some electron microscopy to look at the phenotype of this pollen a bit more closely. And also here, very similar to what we can see in these heat stress plants, we have pollen that seem to be empty and are also smaller in size in comparison to the wild type. And also when we look in the entire spike, so the effect, this once again looks similar to heat stress plants. So in Golden Promise Fast, we have a spike producing nicely a lot of seeds. And in the mutants, we can see a lot of spikes that produce either no seeds at all or only a very low number of seeds. And this you can already also see here in the spike fertility on the right, which is significantly reduced in the mutant plants. And also when we look at whole plant level, so looking at the tiller fertility, meaning the percentage of tillers that produce at least one seed. So the bar is very low, but also here we can see that this percentage is really, really low in the 3 mutants. So this yield potential that these plants have is definitely not utilized. So we wanted to understand the genetic background of this, and for this we also did an RNA-seq experiment in the same genotypes, which you see here on the left. We did this in 3 developmental stages, so during reproductive development, during spikelet induction, and also during floral development. And we did this in 3 different tissues, in the developing inflorescence, in leaf, and also in stem. I'm only going to focus on inflorescence mainly and a little bit on leaf. But to determine differentially expressed genes, we did pairwise comparisons, so always comparing the mutant to the respective wild type to see which genes are differentially expressed. And when we look at the number of differentially expressed genes here in the inflorescence, here on the left, we have the Golden Promise Fast background mutants, and then on the right, the Golden Promise background mutant. The light orange are upregulated, the dark orange are downregulated genes. In general, we can see that more genes are upregulated in the mutants than downregulated. And what we can also see is that in the Golden Promise Fast background mutants, we have an increased number of differentially expressed genes at budding stage 3.5, so during spikelet initiation. So maybe it's possible that this developmental stage is affected most by FD1 and the other ones not so much. And what we also see is that in the FD1C mutant compared to Golden Promise, we see a lot less genes that are differentially expressed. And I think this makes sense because when we come back to the flowering time here, and when we look at the distance between Golden Promise Fast and the mutants and the distance between Golden Promise and the mutants, this one is of course much bigger than when we compare Golden Promise and the mutant. So I think it makes a lot of sense that when we compare these things, we see a lot more genes differentially expressed. And this is also the case when we look at the expression of FD1 itself. So this is in the leaf, it's not expressed in the inflorescence. We see also quite similar to what we saw in the heat stress plants, that we have high expression of FD1 in Golden Promise Fast. In Golden Promise, we have very low expression, which is slightly increased during development, but the overall level is very low. And the FD1 mutants are actually very similar to the expression in Golden Promise. However, if we now look at FD1 targets in the inflorescence, so I also have FD2 here as an example, we can see that it does actually make a difference whether we have a lowly expressed FD1 gene as we have in Golden Promise and a non-functional FD1 gene as we have in the mutants. So here we very nicely see this gradient in the expression of FD2 in the inflorescence. And I'm also showing some other Marzbox gene here, so Barley Marzbox 3 and 8 and a Cibalata-like gene. In all of these genes, you can see this gradient from Golden Promise Fast to Golden Promise to mutants. And these Marzbox genes are actually, as Maria also mentioned before, involved in inflorescence architecture. And when we look at the architecture of the FD1 mutants here, this is a very severe example but I could see at least this to an extent in almost all inflorescences that I looked at. You can see that we have some secondary inflorescences here growing from the position of the central spikelet, and this also resulted in these branched spikes later on the plant. So the downregulation of the Marzbox genes might be an explanation why we see this deregulation of inflorescence development. One more thing I would briefly like to show you is regarding hormones and hormone homeostasis, because the hormone patterning within the inflorescence is very important, and it's a very, very delicate system. The distribution within the spike is quite important, and actually this is how VRS2 mediates this regulation of inducing the spikelet number, so whether a spike is 2 or 6-rowed. My RNA-seq data is of course full inflorescence, so I can't make any conclusions about the distribution of hormones within the inflorescence. But what I could see is that many genes that might be involved in cytokinin, auxin, and gibberellin transport are up or down regulated in the inflorescence. So this has the potential to also result in some faulty patterning, and might also be an explanation why we see this branched inflorescences in the FT1 mutants. And one last thing, so this is also something that Maria touched before, we looked at the micronutrient content of different micronutrients in the seeds of these plants. So one example here is iron that I'm showing, and here we can see that there is also some FT1 dependent effect on the concentration of micronutrients. We can see that the iron concentration in Golden Promise fast background mutants is significantly increased, but in Golden Promise, for example, we don't see a significant difference to Golden Promise. So there seems to be some kind of PPDH1 dependent changes in micronutrient distribution. And when we look at different transporters that are potentially involved in iron and also in zinc transport, we can also see that these are differentially expressed in the mutants. So it is possible that micronutrients are actually somehow differently distributed in the plant, and this could be part of the explanation why we see this reduced yield in the FT1 mutants. So to sum things up, this is what we already knew. We did know that FT1 promotes the shift from vegetative to reproductive growth, but we didn't really know what other effects FT1 had. And actually, it doesn't only affect flowering time, as we already knew, but it affects a whole lot of other things regarding longevity and also plant and spike architecture. It negatively affects yield in the plant on a whole plant level, but also on a spike level. It changes micronutrient contents within the plant, and it potentially also changes hormone patterning within the plant. So FT1 really has a surprisingly pleiotropic effect on development and plant architecture, and this will hopefully help us to further understand the role of FT1 in this really very complex network of flowering regulation and plant development. And with this, I would like to thank my supervisor, Maria, and the rest of the group who are always very, very supportive in my work. I would like to thank Jochen Kumlin and his group who created the CRISPR knockouts for us, and also Stanislav Kopriva in Cologne, who did the micronutrient measurements for us. And of course, I would like to thank you for your attention.
Thank you, Gesa and Maria. Those were great presentations, I have to say. I see already some people applauding. I actually don't know if people put questions in the chat box, but I don't see any of them. So is there any?
There's no questions there.
There's no questions in. Okay. So let's first start taking some questions from the audience. Are there any questions? If you just raise your hand then, or just unmute yourself and speak up, maybe. Can I start if nobody wants? So actually, Gesa, I would first like to have a question for you. Because you present this very exciting RNA-seq data from the FT with many differentially regulated genes in the FT1 mutant. And you show that it's a very conserved network and a very conserved pathway. And I was wondering if you look now at potential downstream targets of this FT1 signaling, how much of that is now conserved among species? Can you estimate this based on your data? If you would compare like known genes, for example, from Arabidopsis or rice, do you find overlaps there?
I'm still working on the RNA-seq data, I must admit, so I'm only at the beginning of analyzing it. But at least when it comes to these ABC genes and so on, this is also something that you can see in Arabidopsis. So I guess this is kind of similar to what we also see in barley. So I'm assuming there is quite the overlap between barley and Arabidopsis.
Okay, and then the more difficult question, what do you think would be the prime differences between? Maybe you can't, it's always difficult to answer that based on the data, right? But yeah, if you already have a indication on that.
I'm not sure yet. I do see some differences in light harvesting complex genes, for example. So we do see some upregulation there. I'm not sure if you actually can see something like that, some photosynthesis related genes in Arabidopsis. So I think that might be something that is different.
Mm-hmm. Great. So are there any questions from the audience still? And can I just go on with questions? I don't see them. It does. Donna, do you see people raising hand? Do I miss them or?
No, I haven't seen any, but if you want to stop sharing, then we'll see everybody on screen. It might be easier there.
Yeah, exactly. Because it would be.
So if anybody wants to ask a question.
I had a question. Can you hear me?
Ah, yeah, great. Yes, now I can hear you.
Yeah, I
go ahead.
raised my hand. Sorry, but I guess you don't see that.
I couldn't see them, so I was like, okay, where?
Okay, yeah, thank you. So I just had a question regarding the transition from the SAM to an inflorescence meristem when you had the two group of plants flowering versus, you know, earlier versus the late flowering. So did you see any changes over the transition at the macroscopic level, you know, from the SAM to an IM? So any kind of, you know, morphological changes over the transition, and how does it might affect on the flowering times and also maybe the production of the seed number?
Is that a question to Gesa or me?
Yeah, maybe Maria can maybe can be
Hey,
related.
can you? I think I haven't fully understood the question. So is that, did you ask whether there are changes in the inflorescence meristem?
Yeah, the transition from the SAM to inflorescence meristem, you look at you show a macroscopic image of the SAM.
Yeah.
So I'm just wondering, have you looked at more details about how any changes over the transition might affect on flowering times and also the number of the seeds?
No, I mean, of course we assume that there are some differences occurring here, but we haven't really looked at this at a very, I mean, at a cell level detail, I would say. So what I've shown is that there are changes in the size of the inflorescence meristem that we can observe, but other than that, this is something that is ongoing. So we're trying to look a bit more closely at what happens also at the cell level, but at this point I cannot tell you.
Okay, yeah, thank you.
Okay, I see a question in the chat from I hope I pronounced this correctly, Jingyun Yu. And the question is if it's known if these micronutrients that you presented, such as iron or zinc, if they affect inflorescence architecture, and if yes, how does that work?
No, I don't know whether that, so what we measured at this point is the micronutrients in the seeds. We have not measured micronutrients in the developing inflorescences. So I think the assumption would be probably the other way around. It would be, so our data suggests that differences in the development and in the morphology of the plant and the inflorescence has an effect on the micronutrient content in the seed. So I would formulate it the other way around. What the effect of this is on the seed viability, seed germination, we don't know. Kumsal in our group, she's actually used seeds and from the heat and the control temperatures to grow them and phenotype the offspring under high and controlled temperature conditions, just to check whether there is a difference in the quality of the seeds that come from the control and high temperature conditions. And so far, it didn't seem that there is a difference. So we couldn't really see any difference in the germination rate, in the seedling vigor, and also in the actually performance of the plant, so this the next generation. But of course, a different composition of the seed can have an influence on the downstream use of the seeds, or for human consumption or animal consumption. I think that could have implications. I will also not exclude the possibility that it has some effects on the quality of the seed in the end. But I'm just saying we could not really see that so far. Not sure if that answered the question.
I'm not sure if it answers, for me it answers it, but I'm not sure if it answers it for the questioner. I do see a second question coming up in follow-up of that. And that question is, do you think if the photoperiod and heat affect male fertility through the same pathway, so both through FT1?
Well, that seems to be the case, yes. So because both photoperiod and, I mean, that's the hypothesis that we put forward here, that FT is quite central. And that's, I mean, this is something that Gesa showed with the knockouts. It's quite central for flora development and flora fertility in grain set. It does have an effect on the induction of spikelet primordia, but you could see from her data that even in the absence of FT1, spikelets are induced. So this is delayed and prolonged, but it's not impaired. What is really mostly affected by FT1 is the flora development. And it seems that high temperature or ambient temperature variation and photoperiod variation converge to some extent on FT. I mean, this is what we see. We cannot exclude that there are, and by comparing our data, of course, we can also see there are some differences between the FT knockouts and what we see under high ambient temperature conditions. So I wouldn't say that all phenotypes that we see under high ambient temperature conditions are completely conveyed through the downregulation of FT1. So here I would be careful. There are probably many more effects. But to some extent, yeah, so the FT1 can mirror what we see under high ambient temperature conditions, or what we see if we have short days instead of long days, because that then downregulates also FT1 and has a major impact on floral development.
Okay, so I still see a hand from Messi. Is that the previous hand, or is that the new one? No answer. Okay, then I'll just go to one of the questions in the chat box, and that's a question from Ender, who asked if the vegetative. So how can this effect that you see of the shorter vegetative growth period in the golden promise fast affect your final yield?
So the question here is whether the fact that there's less vegetative growth, so there's fewer vegetative biomass, whether that has an effect on the yield.
That's how I would interpret it.
Yeah.
Send the question. Yeah.
It may.
Yeah. Yes.
But so what we see is the biggest difference is because the golden promise fast develops faster, so has a shorter period of time of spikelet induction. So golden promise fast plants have shorter spikes because fewer spikelets are induced. So that could look actually negative under control conditions because, as Gesa said, the number of spikelets really are important for what we call the yield potential. But then even under control conditions, the fertility or the ability of the spikelets to produce a floret in the grain is higher in golden promise fast than in golden promise. And here again, we think that is linked to a difference in FD1 expression in both genotypes. So yeah, I mean, it's always been kind of discussed that this longer period of growth in the spring barleys that have this mutation in PPH1 is good in environments with a long season, because these plants can accumulate a lot of biomass and then have a lot of assimilates to pump into the seeds. That could be true, but definitely under a bit more limited seasons, then being fast is much better, because what PPH1 and FT also control is senescence. So it's on both ends of the development. So there is an earlier senescence, and senescence can be positive because that really then makes the nutrients being transported into the spike. So I would say it really depends on the environment, because always having more leaves is not, I mean, having more leaf material is not always beneficial. It really depends on the environment. So I said these golden promise, which are slow, they have more spikelets, but of course, because they have a longer vegetative growth, they produce more leaves, and in each leaf axil, there's a potential of a tiller forming. So typically they also have more tillers and they have more spikes. So I think this is where the yield advantage comes in much more, because they are really having more tillers per plant. But yeah, so that really all depends on the environment. And this is something that we showed by seeing these different correlation of biomass and leaf number on grain number, on final grain number, depending on the temperature. You could see the same if you have more or less water, or yeah. So any other stress, it would probably look very similar.
So I think there's a nice follow-up question on that previous one, which asks if you would now put this this GP fast in the Mediterranean climate, would it perform better when compared to the GP, for example?
Yes, definitely. So if you look at the distribution of the alleles at PPH1, you see the wild type much more common in Mediterranean environments, and the mutated one is more common in Northern European environments. But then if you use these integration lines and put them next to each other in different environments, and this is something that's been done by Klaus Pillen, and also Ayed Abdalati was working at some point with me in Jordan, you can see that the so golden promise fast is doing much better in Mediterranean environment with a terminal stress in summer, and typically golden promise fast would be growing better in Northern European environments.
Okay, thank you. I think that's most of the questions from the chat. I see here an okay, perfect. Indeed, that was a perfect answer. So I have a bit difficulty seeing raised hands. So if you would just please either put it in the chat or just shout out if you have any more questions. Just wait a bit. Then maybe I can still ask one. Just so Maria, you showed this really nice DR5 marker lines for auxin signaling, and you said you were crossing that into your GP-fast lines to make a sort of reporter line to see the effect of altered auxin signaling. And Gesa mentioned briefly that this was also like that in the VRS2 lines. So you're also crossing this, for example, auxin signaling markers with other report lines with, for example, a VRS2, so that you could see, like, make a sort of comparison on how the marker would work.
Well, I mean, here in this context, we are really interested in understanding how temperature and PPDH1 affects auxin, so within this specific project. But of course, it's very interesting to cross that to other developmental variants that we have that are changed in developmental timing, but also in architecture, in inflorescence architecture and whole plant architecture. Yeah, so this is something that is ongoing. Yes.
Yeah, that will be really nice to see to see those results as well. Looking forward.
Yeah.
Yes.
Yes.
So, are there any other questions from the audience? Because it's also nearly time, so if there's no more questions, then I would like to end this very nice seminar, and like to thank to both speakers, Maria and Gesa, for their presentations, and I hope to see you next seminar series.
Okay, bye. Thanks a lot.
Bye.
Bye, thank you.